So today we're going to talk about inhaled biologics, and I'm joined by my colleague, Ben Hatton. So taking a zoomed out view, biologic drugs make up more of the pipeline than ever before. And we're entering a golden age, from cell and gene therapies to RNA vaccines, via monoclonal antibodies and oligonucleotides. In fact, biologics represented approximately 40% of all FDA drug approvals in 2022 and 2023, and sources converge on a biologics market compound annual growth rate of 9 to 10%. Ben, how are these drugs being currently delivered?
So IV is the most prevalent delivery method and very much the starting point for most formulators. But increasingly there is a push towards more convenient methods of delivery. For example, intramuscular or subcutaneous delivery, which is a natural progression from intravenous if the formulation is of sufficiently low volume and can be concentrated without increasing viscosity outside of a deliverable range. Immune reactions, injection site enzymatic interactions, and other similar issues could make this challenging, however. It does present, though, a greatly reduced delivery complexity and cost over intravenous. Then there's the oral route, which is the truly simple administration route and the most widely patient-accepted dosage form. But there is a potential for degradation in the gastrointestinal system, and so far this has precluded most biologics from being delivered via this route. Then there's inhalation, or nasal, what we're talking about today. An established, non-invasive route, widely understood and accepted by patients. There's an ability to deliver a relatively wide dose range, up to 10 millilitres, but there are specific challenges in delivering to the lung. So Svilen, why would we deliver to the lung?
So that is an excellent question. And in my mind, the primary driver is either vaccines or local delivery. So when it comes to vaccines, the advantage is mucosal immunity, which you don't otherwise get through systemic delivery. And since most pathogens enter via the mucosa, therefore you can get the first line of defense through inhaled vaccines. And this is already starting to be tried out, for example, with COVID booster shots. Then there is local and more direct targeting. So lung and heart disorders are obvious candidates that could benefit from inhaled delivery. Lung cancers in particular are receiving a lot of attention recently, and Ben, I know you've been looking into this. Could you tell us a little bit more about this?
Yes, and as of 2023, biologics made up 51% of all approved oncology drugs on the market, and in fact 64% of oncology drugs specifically targeted at solid tumours. And delivery via the lung tries to avoid the systemic toxicity associated with the intravenous route, which involves higher doses and higher levels of side effects as well. Now, due to the stakes involved in oncology, at times these indications afford a more complex delivery route. So lung toxicity in any application is a key concern with drugs delivered via the pulmonary route, and minimising dosage to unaffected regions and verifying that dose has been correctly administered to the tumour can be achieved with more interventional strategies, including drug delivery add-ons to pulmonary endoscopes, for example. This also recognises that with select therapies, the high cost per milliliter, lower doses mean lower toxicity and a lower overall drug costs. Now, as with all routes, this is a good example of how it's highly contextualized on the drug. With some cancer and drug types, the systemic effects of an IV route are actually desired, as they act to prevent or mitigate a metastatic progression, for example.
That is an exciting space, no doubt. However, it's not just about mucosal immunity or local delivery, is it?
No, there are two other advantages off the top of my head that delivery to the lung enables, one of which is that it's fast-acting, and secondly, it's non-invasive. So lung delivery is generally fast-acting, and we see recovery products such as asthma rescue inhalers leveraging that fact. But equally, it's fast-acting in systemic delivery as a very efficient exchange mechanism into the bloodstream. So, for example, Narcan as a recovery product also leverages that benefit. And equally, there's other applications in post-exposure to biological agents, whether that be defense and security applications or industrial accidents. Equally, it's low or non-invasive. Systemic delivery can be achieved, we know, with smaller biologics such as peptides and proteins, monoclonal antibodies, and the parenteral route has its own challenges, from needle phobia to infection risk. the latter being especially important in developing nations. And equally with powders that require at delivery resuspension for injection, this brings a specific set of challenges of its own with variability and homogeneity, etc., which we've covered in a different video.
Yes, and these advantages, of course, present exciting opportunities. And so it's worth contextualizing what the inhaled biologics market currently looks like. So at the moment, there are only 2 approved biologics. So that's Dornase alpha and Afrezza. And there are about 20 molecules in various stages of trials, mostly in the form of nebulized liquids. And most candidate drugs are to treat lung conditions such as cystic fibrosis, RSV, asthma, COPD, and of course, lung cancers. And at the same time, there are a few other applications being investigated, including treating multiple sclerosis and ricin intoxication, for example. So for me, the obvious question then is, why haven't inhaled biologics massively taken off yet?
So looking at it very simplistically, delivering to the lung via inhalation is much more challenging than delivering systemically via an injection. Mm-hmm. When you inject, you rely on a needle to overcome the body's barriers to uptake that drug. With inhalation, however, you rely on a device to generate droplets of the right size that get to the right place in the lung. And then on the formulation to make it through the mucosa, the epithelium, and the other barrier mechanisms that the lung presents. This means that there are multiple challenges for inhaled delivery. Perhaps you can go into more of those.
Absolutely. And the 2 challenges that come to my mind are around delivery and formulation. So if we talk about delivery, there's of course this complex airway structure that you need to navigate. And this requires precise droplet sizes of 1 to 5 microns for deep lung deposition. Then there is the presence of defense mechanisms like mucociliary clearance and alveolar macrophages that can trap or remove biologics, reducing their therapeutic efficacy. Then the deposition and efficacy is affected by lung diseases. For example, COPD can alter the airflow into the lung and the level of mucus. And biologics for systemic effects need to cross the lung epithelium, as you mentioned, and enter the bloodstream. So factors like molecular weight and receptor-mediated transport mechanisms influence absorption. Now, looking at the formulation, there are constraints there too. So the range of FDA-approved excipients is limited, restricting the options to stabilize biologics. And also different excipients may be needed than for parenteral. So some may interact poorly with the device. For example, there might be foaming or high shear forces. And we need to be careful how the formulation interacts with the lung when the formulation lands there. For example, pH, polarity, etc. are all important considerations. And lastly, there's of course the immunogenicity concerns. So inhaled biologics may face a higher risk of triggering an immune response compared to other administration routes.
So to continue with that list, for example, the dosing regimen and quantity and response with the delivery to the lung is going to be different to a systemic delivery route, and this requires additional clinical trials to establish what that difference is. For example, with RNA-based COVID vaccines, certain studies suggest that 100-time reduction in dose may be possible relative to a systemic delivery route. Then we have to consider that doses for therapeutic effect are larger than typical doses delivered by most existing inhalers. There's also degradation issues, so certain biologics are susceptible to degradation, such as protein unfolding, aggregation, loss of encapsulation efficiency in an LNP system during the shear stress involved from aerosolization, manufacturing and storage. Manufacturing and storage are also concerns that are common to injectables, but are potentially amplified by the particle sizes that we're trying to achieve in inhaled delivery. This is also dependent on formulation, but a shear stress of 100 kPa may start damaging COVID vaccines, which is well within the realm of an inhaled aerosolization method. There's also considerations of the past. So, for example, we all remember Exubera, a product with an incredibly high development and actual cost. There are also concerns with this product on side effects such as lung carcinogenicity. Patient acceptance and usability challenge amplified the issues with that device, and I think that pharma may be reticent to continue with biologics via the inhaled route, potentially due to this problem. So all of these challenges place a lot of constraints on the delivery device itself. They need to produce the right droplet size, be compatible with and not significantly damage the formulation, and also have the right usability elements and cost. They must support the right dose, volume, and ensure that there are no start-up and transient effects during the aerosolization process. So Svilen, maybe you can talk to some of the options that we have in devices.
Sure. So if we go down the list of device considerations, starting off with, for example, dry powder inhalers, they require overcoming additional manufacturing challenges relative to, for example, using an aqueous formulation, because you need to produce a dry powder. And also DPIs may not be able to deliver a high dose, so typically they're limited to less than 100 milligrams. Then you've got PMDIs. Their formulating is difficult. Proteins are hydrophilic and non-soluble in typical non-polar propellants, and it's difficult to deliver a high dose. Then, of course, there are considerations such as, for example, thermal shock that may occur due to expansion cooling, and there is the potential of causing damage. Now, you've also got soft mist inhalers, for example. This is typically done with impinging jet technology, and it requires high pressures, about 250 bar, which can lead to very high shear stresses and limits the dose volume given the power source requirements. And also, SMIs impose quite a low throughput, so we're talking about 10 microliters a second. Lastly, you've got electronic nebulizers, and there are several categories. There are jet nebulizers, vibrating mesh nebulizers, surface acoustic wave nebulizers more recently. And at the moment, it's mostly vibrating mesh nebulizers that are preferred, as they're readily available, they can accommodate a wide range of dose volumes, and vibrating mesh is least damaging. But these devices are bulky, they may be complex to use, they require a power source, and also they generate heat, which may damage the formulation if large volumes are being delivered. Not to mention that the shear stresses can also be high.
Yes, and whilst in the electronic nebulizers category we consider vibrating mesh to be the least damaging, an extension to the nebulization category would be Rayleigh breakup, mechanical route. And this operates at a lower pressure than most inhalers, so less than 20 bar, and shear stresses are also a little bit lower. So we're also talking about lower heat damage from the mechanical route, because we're not getting the same heat accumulation. However, there's not no damage. Studies do suggest that it can reduce encapsulation efficiency of formulations like LNP encapsulated RNA vaccines, largely due to shear stresses. Operating at a lower pressure also means a lower particle momentum. So there is a challenge in reducing deceleration of a particle and induced coalescence. So airflow needs to be carefully considered to accelerate these particles away from each other. It also may be challenging for users to use if we're delivering large volumes in a solely user-driven mechanical system, rather than something that might be electromechanical, for example.
Innovation will no doubt be needed to enable the inhaled delivery of biologics. And it may be tempting to think about innovation as a sequential process, or one that focuses on a core area, for example, a great formulation. However, it is also clear that inhaled delivery of biologics is a tightly coupled challenge, and upstream choices can lead to costly or difficult to achieve downstream requirements. The aerosolization science to generate a plume with the right characteristics, and the device engineering for a design that is suited to the use case will also be key. So, in my mind, the development journey for an inhaled biologic therapy is really like a game of snakes and ladders, or chutes and ladders if you're from the US.
So, like snakes and ladders, the path from start to finish is fraught with potential setbacks, your snakes, and shortcuts, your ladders. You roll the dice, and sometimes you're lucky to catch a ladder. For example, we find a device that does not damage your formulation, or a formulation that we start with is ultra shear resistant. Sometimes, however, we're unlucky and we land on a snake. For example, you find out your device only works if you deliver very small amounts of formulation, because we see heat accumulation and that damages the drug itself. And then you're back to square one. And so the process repeats.
That's right. And I think we can do better than just roll the dice. We can significantly bias the dice in our favor, so we can get to the end goal faster and with more certainty. For example, if we knew what boundary conditions, so think shear stresses, pressures, heat and power inputs, each mode of aerosolization imparts, then we can assess earlier how a candidate formulation can perform. We can understand this early through experimentation, experience, modeling, and of course a tight collaboration with the formulation teams.
So if we understand what thresholds for damage are and what the levers are available in the aerosolization technology, then we can tune it. For example, shear stresses on a vibrating mesh can be reduced by increasingly piezo coupling efficiency and also altering the mesh hole geometry, to name a couple of methods. And maybe with the right tweaks, a vibrating mesh could be the right aerosolization method for your formulation. We've developed over 30 inhalers and are familiar with each of the major aerosolization technologies, as well as several experimental ones that have not yet been commercialized. We therefore recognize that biologics come in all shapes and forms, and each formulation will have a specific device need. The formulation is the primary value add, and getting an inhaled biologic to market is a couple challenge. There is no one-size-fits-all solution, and so it is important that we remain device agnostic in this process.
Absolutely. And I think the best solutions are the ones that minimize risk and only innovate when absolutely needed. And I suppose on that note, we look forward to helping our clients shortcut the journey by asking the right pragmatic questions and using our deep understanding of the fundamental science. And to avoid the pitfalls by leveraging our many years of experience of developing devices for inhalation and identifying where innovation is and isn't required.